Okay, good afternoon, everyone. Let me confirm again, is my voice fine? And you're able to view my screen?
Yes, sir.
Okay, good. So we'll continue with the chromosomal theory of heredity, right? We will do that first. One second. Yes. So this is what we are trying to learn. So we did learn about Mendel, and we learned some of the terms: a gene, what is an allele, a trait, what is genotype, and what is a phenotype, homozygous and heterozygous, what is dominant and recessive. We also have seen some of the things about what color would happen. I think we also discussed, we had a discussion about dominant and recessive. So this is about the principle of independent segregation. What you must remember is also this: the essence of try to understand what is independent segregation. It is about talking about the alleles. Each of the trait is coded by two alleles, right? And those two alleles could be homozygous or heterozygous. Homozygous is something like RR, or for example, small r, small r. Heterozygous is capital R, small r. So when we have either homozygous or heterozygous, if a cell is, for example, heterozygous, and it is going to make gametes, capital R is going into one gamete and small r is going into another gamete. That means to say the genes or the alleles are segregating independent of each other. That is called as, that is the principle of, or the law of independent segregation. I'm not going into the details of the Punnett square and all. We are all familiar with that. So all I want you to remember is independent segregation. It's about the alleles segregating independently to gametes, which will fuse with other gametes from maybe other organism and form a new offspring. And we also discussed about dominance, codominance, where both the characters are expressed. And here I only want to talk about what is meant by independent assortment. Say, for example, we are talking about two traits. That is when we talk about two dihybrid cross. That means in the cross, we are evaluating two different traits. Say, for example, we have RRYY, that is for round and yellow. Then the offsprings, all of them that would form, if they were, all of them were RRYY, irrespective of small y, say if there is a cross between an individual like this and like this, and there were 50% of the individuals are of capital R, capital Y, and 50% are small r, small y. What does that mean? It means that the assortment, the characters are not assorting independently. But what did Mendel find, or anybody who carefully examines, is that the offspring is usually a mixture here, right? And if you see in the F2 generation, the mixing is much more profound. And you will get several ratios, phenotypic ratio and genotypic ratio. And you don't have to mug up the ratios by themselves as such. Rather, I would want you to be able to determine by drawing a Punnett square. It might take a little while, but you will never go wrong. So learning facts is something, learning principles is something. Facts is something like 2 + 2 = 2. equal to 4. That is a fact. So 2 plus 2 equal to 4, or I think it's much more obvious when we are children. We are made to mug up the tables and say 6 into 5 is equal to 30. Right? We are made to mug up these things. We are learning facts. But what if you knew how to do 6 into 5? Make 6 groups of each containing 5, and then you be able to determine that it is 30. That is learning the principles. By hearting or mugging up the facts is easy. I mean, it will save you time. Whereas if you are learning how to determine it, you can use the same principles for many other things in the future. So always try to learn the principles properly so that you will be able to use them in your future anywhere applicable. That it becomes learning of principles is it becomes part of intelligence immediately. Okay, I hope you understood what is meant by independent assortment. Okay, there is a problem. Oops. Sorry. Yeah, this is where we are at. Okay. So there is one another example in the textbook. I would want to rather focus on that, which there will be more details, and it's about Drosophila. It's a typically used organism in genetics, classical genetics, and that's why I would want to talk about that. So here is a female. This symbol is for female. Okay, 1 second. Oops. Sorry, just give me a minute. I hope you can see it good enough. So here is a female. This symbol, I think you know it. It is for female, and this represents male, right? And then the female has, there are two things in this particular diagram. What we are going to see is whether there are two things. That is, one is sex of the individual and the eye color. Okay, sex and eye color. Let me write it clearly, something is wrong. Sex, whether it is male or female, and then it is eye color. Okay. So here is a female with red colors, red eyes, which is for some unknown reasons it's given as WW. The opposite would have been the white eyes. Okay, red female, red eyes female, white-eyed male. If there was a cross between these two, then there will be something else. The individuals will form, they can form either of them. Red-eyed female and also red-eyed male. So the red is, having a red color is dominant. Okay. Although the individual got capital W and small w, because capital W is the dominant and it represents red color, that is being exhibited in this female. In the male would have had Y. Okay, here is something that you need to understand. I will describe that in the later slides as well. If you don't understand here, that's all right. Here, Y representing, it is something like females have 2 X chromosomes and male has 1 X chromosome and 1 Y chromosome. For simplicity, in this particular one, they did not represent what is Y. Okay. If it is Y, it doesn't have the particular gene for red color is present, for eye color is present on X chromosome. Okay, the W that is being shown here, that is present on X chromosome. So both of them are XX here. Whereas in Y chromosome, there is no eye color gene, it is lost. Okay, so where does the eye color come from in a male? It comes from the X. Something is a little bit troubling, but I hope I'll be able to make X and Y. Just give me a minute. I need to check something is pen is not writing well. Just be online, please. Yes, can you hear me?
Yes, sir.
Okay, so there is some issue, but I'll try to make this up in a better. I'll record a video and I'll post it later, maybe. But otherwise, right now, I think one will require, if you're reading for the first time, one would require to listen to it twice. So it's about X-linked inheritance, right? Just try. Let's see if we get through. Otherwise, we will do it again. So here is X and X, and the male would have X and Y chromosome. Okay. As I said, first things what we should remember is X chromosome has the eye color gene. Okay, eye color is on X chromosome. So the female would have had two X chromosomes. So there are two alleles for eye color. One is capital W and the other one is small w. Red color is the dominant phenotype. Therefore, you will see that the individual has red eyes. In males, it has one X chromosome and one Y chromosome. And if it has inherited, it will have to inherit one X chromosome from here, and Y chromosome will come from the father. Therefore, the eye color is determined only by the X chromosome. So it would have, because it's capital W, it will get red eyes here. Now, let's go a step further. Here, when the female forms gametes, it will have capital W and small w. And when the male forms gametes, one X chromosome, this will be the X chromosome, this is the Y chromosome. Y chromosome does not have the eye color gene, whereas X chromosome has. So one X chromosome from here and another from this one would give rise to female. And this is also another female from this X chromosome and this one, this gamete and the other gamete. So you will get female two females with red eyes. Whereas in the if it is a male, if this gamete fuses with the Y here, then you will get a red male. If the other one, this W, the small gamete containing the small W, and the Y gamete with the X chromosome, if they fuse, they will form a white-eyed male. Okay. So just try to remember it that way. If not, I will try to post a video shortly. The second one is also very much similar, except that the parental genotypes are changed. And the genotype is, here we started with red female, whereas here we are starting with a white female, a white-eyed female, and red-eyed male. That is opposed to white-eyed male. So everything will go according to the same way. What you should understand here is that the eye color is linked with X chromosome, right? So there are several traits even in humans that are X-linked, and that we will discuss maybe in the next class. I'm unable to write properly. So in independent assortment that we have learned here, what we have learned is that chromosomes, the traits are assorting themselves independent of each other. And the molecular basis for it is linkage, and the point is the crossing over. Where does crossing over or synapses or chiasmata? It happens in meiosis. In meiosis, they form homologous chromosome pairing. When you say homologous chromosome, that is after G2, that means each of the chromatids has duplicated each of the chromosomes. So there are two different DNA molecules attached together, something like this. And there would be another one, homologous chromosome. When I say homologous, what does that mean? It has the same genes on either of them. There could be little bit of differences in or mutations, but otherwise the gene sequence and the nucleotide sequence is almost similar. So here are two homologous chromosomes I have drawn, and these two come and pair together. Let me get this straight before we progress. These two would be called as chromatids, right? They are sister chromatids. Whereas if you talk about this one and this one, they are non-sister chromatids. Right? They have same order almost, but there is some difference. So they are non-sister chromatids. What we should notice that crossing over almost always happens between non-sister chromatids. That we have discussed. If it happens between sister chromatids, there is no exchange of information. It is just waste of undergoing all that process. So as you can see here, the chromosomes, this is one chromosome, this is the other chromosome with their sister chromatids, and the non-sister chromatids will undergo crossing over, something like this. Some form of physical exchange of chromosome fragments between these two happens, and afterwards they cleave off, something like this, to form exchanged parts like this. Okay. That is how crossing over happens. What happens during crossing over? Exchange of genetic information or the physical parts of the chromosomes happens between non-sister chromatids of homologous chromosomes. That is how it should be said. This is one of the important contribution by McClintock and Crichton is they have proved that there is the exchange between these two happens as a physical part. They have used a different experimental system to show, I think it is Drosophila, where one of the chromosomes had an extra piece like this. So if crossing over happens between them, what would occur then, right? will this be going to the other chromosome or not? What they have observed is it's always that this whole part, WX, small w, small x, always goes with this extra chromosomal material, right? Anywhere you see, it's always linked to it. I think I should also tell you what is meant by linking, gene linkage. So I will try to draw a chromosome here. This is a chromosome, and this is gene A, gene B. Something is bad. Okay, gene C and gene D. And I'm going to draw another chromosome, a different one, not a homologous chromosome. Gene E, F, G. Now I'm going to say some sentences, and I want you to understand what would it mean in terms of genetics. So if I said gene A and gene B are linked, what that means is they are present on the same chromosome, right? So if gene is being when they form gametes, you will often see that A and B will go together. Why is that? Because they are on the same DNA fragment. Okay, just I'm trying to define what is meant by linkage. Okay, don't take it further. We will see if required in the next classes. So when two genes are present on the same chromosome, they are linked. If gene A and gene E are not linked, what does that mean? That means they are present on two different chromosomes. Now, there is one more that I have to say is that we can also make some more comparisons. Gene A and gene A is more linked to gene B than it is linked to gene D. Of course, gene A and gene D are linked. Gene A and gene B are also linked. But which one are linked very more? You'll find that A and B are very close. Physically, the genes are present very close by on the chromosome. So you call that as are highly linked. Okay, this one, gene A and D, is called it's kind of low linkage. This is what I want to use. This is how you can try to say a few things. Say, for example, there is a chromosome, right? And I'm trying to draw what is already given here: capital A and capital C and capital B. Here is small a, small c, and small b. Okay. Assume that there is one probability of one crossing over happening between these two non-sister chromatids. What is the probability of happening? Say, in one case, it may happen here. In another case, it may happen here. In another here. We are just saying only one is probable. But if you had these, the chromosomes, you had some 6 different sets are there, what will happen? So if a crossing over happened, I'll call this as 1, 2, 3, 4, and 5. So there are 5 crossing overs on different chromosomes. Okay, for my simplicity, I have drawn it here as it happened in one chromosome. So I will draw one of the chromosomes. What would happen if crossing over happened? Then this will be capital A, and this should be capital C, and this should be capital B. Here would be A, small c, and this will be small b, if crossing over happened like this, right? Now, what you should do, oh, sorry. Now what I'm going to do is I'm going to erase this part and join them. What will I get then? I have A, small c, small c, and small b. Small a, small capital B, and capital C and capital B. Recombination has happened. Crossing over, because of crossing over, these parts were exchanged. Now let's also try to do the same for 2, 3, 4, 5. We are not going to draw lines, right? What would happen then? Capital A, and this is the C, capital C, and what would have happened if exchange has happened like this? It would have become small b. And the other chromosome would have been C and capital B. Right? This is for crossing over 1. This is for crossing over 2. For 3, what happens? Capital A, 3 is here, right? So it will still be the same way. A. Okay, I'm having trouble with the graphics pad. Maybe next time we'll do something else. So what you will see, notice that is, as the space is more, the space between AC and CB is more. So what is happening? There is more probability of crossing over occurring between C and B. Therefore, they are likely to find different combinations. Okay, we'll try to watch it on some animation someday, and I hope I'll try to share it, and you can probably see that there. So I'll get to the figure here, and what you should remember is that the more distant the chromosome, the genes on a chromosome, the more probability that crossing over would occur between this space, right? If it is less space, then the probability of a crossing over happening is very less. So when the crossing over is very less, probability of crossing over is very less, then you will say that A and C are highly linked. Okay? Linkage is that. When you say high linkage, that means they are physically very close by. When there is low linkage, then that means they are on the same chromosome but distantly present at distant portions. If there is no linkage, that means they are present on different chromosomes. So this is the same thing. Assume that if no recombination no crossing over has occurred, as shown in here, then it will remain like this. Majority of the crossing over is not a high frequency thing that happens. It happens 1 or 2 times in a chromosome between 2 non-sister chromatids during one meiotic cell division. So if a crossover happened here, so you will see that you'll find capital A, small b, small c, small b. I mean, you see that the chromosomes are exchanged. The blue color, it is indicated clearly here. So there will be about 7.5%. There is also a probability that the crossover can happen here. In that case, you will have 22.5%, because the crossover can happen here, and also happen here. This distance between C and B is very, very high. Because of that, the probability of recombinants in that case will be very high. There is also very rare probability that 2 crossing overs happen in between 2 non-sister chromatids in one meiotic division, something like this. In that case, you will have capital A, small c, capital B. It is twice recombinant. Compare with others, capital A, capital C, and small b. You don't see patches like this exchanged. If you have two crossing overs, then there will be exchange of and the formation of something like patches like this. When we go into when we have a dedicated lesson on homologous recombination and also site-specific recombination. There we will learn the molecular mechanisms of how things happen. And at that time, we will also discuss about the recombination frequency and other things. For now, try to remember these, some of the terms. You can also read the textbook. The illustrations are good. What I want you to now do is, I want you to answer these questions. If you already followed the videos that I sent and you also followed the classes so far, then you should be able to do these things. I'll read out the question. In a dihybrid cross, that means we are talking about two traits. In a dihybrid cross involving two dominant independently assorting traits, round and yellow. So the dominant would be R and yellow is the seed color, around is the shape. Which follow classic Mendelian principles of inheritance. A researcher found 809. If you have a paper and pen, please try to do it along with me. It will be nicer. Plants that exhibited wrinkled and green pods. So wrinkled is smaller, and wrinkled and green is small y. So I hope, should we be doing the whole Punnett square? I want you to come up with this. I want you to tell me the answers for it. Approximately how many plants those are genotypically homozygous for round and yellow pods? You should be, I have given a number that are these. What would you expect? How many plants would you expect to have this genotype? That's the question. Please do it faster. If you can do it, it'll be nicer. Did I say which? That should be in the F2 generation. You're going to draw something like this. Okay. In this, if you know, if you see that how many are RRYY, that's simple way. I'm finding easy way for me. One is 1. What we will do is I want you to understand the ratio between these two. So we'll try to do how many are small r, homozygous for small r and small y. 1. Can you find any other? There is none other. And if you also see for capital R, homozygous for capital R and capital Y, you will find 1. This is kind of trick question, isn't it? It's simple question, actually, not trick question or anything. If you had 809 plants of homozygous recessive, they are recessive, right? Then you would also get something like same 809, approximately 809 plants of with homozygous dominant traits. That's a simple one. If you know the genotypic ratio, it will start with, if you are representing each of them, then you will see homozygous dominant and homozygous recessive. So that's what you will get there. There are some more interesting questions. I would want you to find an answer for this. We are now solving all the questions that we have discussed so far in previous classes also. A scientist has isolated genomes from two different types of phages, phage 1, phage A and phage B. Lengthwise, genomes of the two phages is similar. However, when checked for absorbance, while their concentration was maintained constant, phage B had higher absorbance than phage A at 260 nanometers. What do you think is the answer? Anybody? Anyone who might try? In one of the lessons on DNA, we have discussed about hyperchromacity. Anybody can tell me what is it? Hyperchromacity. It is to say that single-stranded DNA absorbs more compared to that of double-stranded DNA. So when you take equal amount of DNA, single-stranded and double-stranded, as described in this question, phage B had higher absorbance. That means the most likely thing is that phage B has single-stranded DNA. And phage A relatively is supposed to have a double-stranded DNA. It's about how you associate all the knowledge that you have learned so far and think quickly. This is a question based on replication. This is 10 to the power 9. Okay, while copying. The size of the genome is 10 to the power 9 kilo base pairs. The rate of polymerization of one DNA polymerase is 1000 base pairs per second. The time taken for one round of replication is 2 hours. Calculate the number of polymerases involved in replication. That requires your proper visualization also. And I said kilo base pairs. That means you should add, that's something like 10 to the power 12 base pairs. You have to make 10 to the power more 12 base. You have to form these many. So each polymerase would take about 10 to the power 3 per second, right? So that will be about 10 to the power 9 seconds. And then you convert it into hours. I mean, in 2 hours, if it has to be done, how many DNA polymerases should work to do it? In your 10th class or maybe later or earlier, you must have learned there is a brick, there is a wall. One person would finish it in 10 days. If two people work, they would have finished in 5 days and so on. There is how you put in reciprocal or proportionality. How would you use that? So you are supposed to use same things in these kinds of questions. I'm not going to calculate the whole thing for you, but I hope you know that if you want to finish, replicate all of these 10 to the power 9, if it is one DNA polymerase, it would have taken 10 to the power 9 seconds, right? But convert it into hours. If you had 60 into 60 into 2, okay, 2 hours, how many DNA polymerases are required? That's the question for you. Okay, if it is one DNA polymerase, it would take this much time. If it has to be finished in 2 hours, how many DNA polymerases are required? Obviously, it will be more number of DNA polymerases. You are supposed to do the calculation on that. I will skip this question And I want to quickly go to one other question that I had. Just be online. That's the words, that sentence is not being shown here. For some reason, there is an error. Just stay online. I'm coming here. Okay. Can you hear me? Okay. Yes?
Yes, sir.
Okay. Here is a question that I will not discuss ever again, but it might come in the CIA if there or in the end semester. There is a specific way of how you write things in the exam, right? Or represent different things in molecular biology. So the question is about how carefully you're reading in scientific standards. The question is, please read the following sentences carefully. There are several mistakes. Some of them are simple and some of them are critical. Identify and explain each of the mistakes with justification. Then rewrite the whole paragraph accurately. The first thing is never represent DNA like this. Not even when you're chatting or even on WhatsApp. You should always write it in capitals. It's an abbreviation for deoxyribonucleic acid. So you should always put the same thing like this. And here is the similar answer, a similar problem as well. It should be DNA, capitals. DNA is likely the first replicating molecule to have evolved. That is unlikely. It is supposedly the RNA is supposed to be the first or the first replicating molecule, because RNA can also function as ribozyme. Right? There are RNAs that are made up. There are enzymes made up of RNA. So people think that that is possible. Most likely that RNA had versatile functions, and because of which they think RNA is the first molecule, first replicating molecule. It is thought so because DNA could act as genetic information. RNA can also act as genetic information. So this argument is not so strong. Michelson and Stahl have proved that the chemical nature of the transforming principle is ribonucleic acids. These two scientists did not do that, right? Who are there? McCarthy and Avery, McLeod, McCarthy, and Avery. Those are the ones who have studied the chemical nature of transforming principle, Griffith's transforming principle, and they showed that it is DNA and not RNA. Experiments on bacteriophage using radio-labeled nitrogen showed that genetic material is made up of protein. They did not use nitrogen. You cannot use nitrogen because DNA and protein have nitrogen, right? They used radio-labeled phosphorus and sulfur. And they showed that it is made up of DNA and not protein. This kind of exercise is very crucial for you because you will then know how to carefully read something. So in one of the exercises, we have an assignment for 5 marks. Okay, I'm going to give a page with littered with lot of mistakes. You are supposed to find out all these mistakes and explain why those are mistakes. Okay, so that is how you should be able to see that. RNA has 2' OH, but DNA has 2' H. That is fine, I guess. Is that right? I think it is all right. RNA has 2' OH, whereas DNA has H. That's not a problem, I guess. Thymine has 5-methyl group, but uracil does not. 5-methyl uracil is thymine. That is also fine, I guess. But the problem is I said 5'. It is not 5'. It's 5th carbon of uracil. When you say prime, it is actually representing the ribose. So that's the mistake there. If we take a single-stranded DNA and single-stranded RNA of 1200 nucleotides and of the same with the same nucleotides and same sequence, except for the standard differences between RNA and DNA, deoxyribose and ribose, and also the thymine and uracil. And they have about 40% GC content. It is a problem that we have already done. So I would want you to think critically and try to do it by yourselves. It is concluded that each of these molecules have 30% adenine. So you need to check out if that is right or wrong. GC content is there, so you need to check that. In a separate experiment with about single-stranded DNA and single-stranded RNA, we said single-stranded, so this is unlikely, right? We don't know. We cannot be sure about it. And ssRNA, as described, it is chemically determined that ssDNA is composed of 25% T, and ssRNA is composed of 25% U. Based on this information, I performed blender experiment, and I conclude that ssRNA is heavier than ssDNA. You cannot do blender experiment for it. Blender experiment is done by Hershey and Chase to determine the genetic material of the bacteriophages. If I want to see, if I want to have ssRNA and ssDNA, which one is heavier, I might want to go with density gradient centrifugation, so that I will know which one is heavier and which one is not. Okay, even if it is, you can still know which one is heavy and which one is not heavy. Anyway, I think next time we will try to have the class from college so that we will have our own setup there. Any questions so far with this? With these, I'll